Hello and welcome to this webinar titled Applying Low-Frequency Raman to QBD in Pharmaceutical Development, hosted by Biopharma Asia magazine and presented by John Wasylyk, Senior Principal Scientist at Bristol-Myers Squibb Company, and James Carriere, Product Line Manager for Coherent's THZ-Raman product line. My name is Stephen Edwards and I'll be your moderator. Now, please allow me to introduce our first presenter. John Wasylyk, PhD, is a Senior Principal Scientist at Bristol-Myers Squibb Company. He leads a vibrational spectroscopy group who are focused on developing non-invasive, sustainable methods designed to enhance productivity and generate process knowledge. The spectroscopy-based methods have been deployed in areas ranging from early scale-up to manufacturing environments at various BMS sites. The developed methods encompass both small molecules and biologics. In addition, the group provides hands-on training and oversees open-access spectrometers for chemists and engineers. John is currently the marketing chair for the Federation of Analytical Chemistry and Spectroscopy Societies and Pharmaceutical and Biopharmaceutical section co-chair for SciEx 2019. I'll now be handing over to our first presenter. Welcome, Dr. Wasylyk.
Thank you very much for the introduction. Good morning, good afternoon, and good evening to everyone that's online. Today's discussion or presentation will focus on low-frequency Raman and how it's applied at my company in order to control and understand the crystallization process and ultimately make good final active pharmaceutical ingredients, or API as we refer to it as. Why is crystal form so important? And if you think about it, the main driver is its impact on solubility and bioavailability. Having the right crystal form is critical towards bioavailability. So we have to have total control of the crystal form once we make it and get it ready for marketing. Stability is obviously another important factor. You want the most stable crystal form possible, simply because it's going to have to be transported, milled, moved around, exposed, etc., in bottles, and so on. So stability is important. So you want a crystal form that's very stable. You want something processability. And what I mean by that is, let's say you have good, for example, flow properties. So if you're going to put it in a roller compactor, it can flow into the system and through the gates and into the compacting area. So you want good flow properties. You don't want sticky crystals or odd sizes. So you'll probably have to go through some milling to come up with a uniform particle size. And of course, one of the backbones is the intellectual property, the patentability. So you want to get all crystal forms that are available. You want to have them fully characterized, and you want to have absolute control over that crystal form when you're making your active pharmaceutical ingredient, that which will go into your tablets. So what are the common ways that we look at crystal forms? We use differential scanning calorimetry, solid-state NMR, and powder X-ray diffraction. These methods are great. They're the backbone. When we build a model, a chemometric model, or some way to monitor our control parameters, we rely on our primary methods. And these are the primary methods. They're the backbone. The only catch to it is if it's not a release method and instead it's a production environment that you're concerned with, these methods take a long time and don't have an advantage of being able to work in a fast-moving production environment. They're not in line. They can be problematic in terms of impurities. There are varying sample sizes that you need and long analysis time. And if you're doing a crystallization that may take a long time, but you don't want to have it sitting in the slurry or in conditions waiting for results that may take hours or even a half a day or a full day, then you risk breakdown, decomposition, impurities starting to grow in your mixture, in your slurry. So you want to avoid that. So that's why we go to other techniques such as vibrational spectroscopy. So now, vibrational spectroscopy covers a wide range. We do know that we get a lot of information on the long-range order, or we have a crystalline material with mid-IR and near-IR. With Raman, we can also look at polymorph differences in the crystalline matrix, many times in the fingerprint region. That gives us a lot of information, but now we extend that further into the low frequency range. Now, low frequency in the 0 to 200 reciprocal centimeter range was typically not easily attainable many years ago, simply because of the broad laser line. So you're at that cusp between the Stokes and anti-Stokes region around 0, where the laser line flooded or saturated your detector. It was very difficult to pull out information. But with the advent of new filter technology, new applications, we're able to actually use that near the laser line by suppressing it and taking a look at the spectral response. Advantages, well, one advantage is it's much more sensitive, about a 10-fold increase in sensitivity. We see typically, in some cases, what looks like 10 to 12 times the amount. So it's much better than what we had anticipated, which is great. Another advantage is that we can look at amorphous material. There are some cases, and I have an example where you could see amorphous in the in the fingerprint region or towards a higher frequency, but the main player to look at amorphous is in that low frequency range, and I'm going to go into detail on that. And there's a lot of literature on discussion on why you actually have this amorphous specific response. What is important here is that if we have the potential and actually observe at any point during our development cycle an amorphous state, we have to fully characterize it, understand it, and have control over it, because we can't have amorphous, which are less stable, in our crystalline API. So it's very important that we do control that, and that's governed by the regulatory agencies. It impacts bioavailability, so it's extremely important. Now, what is low-frequency Raman? Well, there's phonons that are given off. They're the quantized coupled oscillatory displacements that occur in the crystalline matrix. And this includes the vibrational and rotational vibrations. They're constrained by the crystal size, but they're constantly vibrating, and that is essentially what we're going to be looking at with the low frequency. What gives it a big advantage, as I mentioned, was this amorphous material, or being able to look at it and quantitate it. And this is just a quick example here where we have an active pharmaceutical ingredient with many forms. And I pulled out a number of spectra. Some are the undesired crystalline form, that which had less desirable powder properties, bioavailability, solubility. We have a desired form, and we've run multiple lots of it, because obviously you're trying to produce that. And we also notice that there's a amorphous material. Depending on your crystallization protocol, you can get some amorphous material. And that is referred to here as the boson peak. So I'm going to go into a little bit of detail on that, just to give you a flavor of some of the complications and the current discussions on amorphous states. Now, the boson peak, when you consider this elongated, that's that very broad, poorly defined spectral band in the low frequency range, in that vibrational spectra, it's typically observed, as I mentioned, in the low energy areas. The vibrational density of states, when we're taking the spectra, is proportional to the frequency squared. But in some cases, when you normalize that vibrational density of states, sometimes called VDOS, by the frequency squared, you get a differentiation or a deviation from the Dubai law. And that's what appears to arise in this boson peak, this elongated spectral band. So in the crystalline lattice, and this is some of the current discussion, and I have a really good reference there that's very up to date on new discussions on what gives rise to it. And a lot of the feeling is it could be in that crystal lattice, there is actually a competition between the elastic mode propagations and diffuse dampening. So there's a lot of vibrational changes and competitions going on when you're in this amorphous state. Now, traditionally, I know it as what Felix Franks would say, it's random, it's ordered randomness, or you've got a small amount of ordered material, crystalline material, that's embedded in a random area. So you don't have enough to have a fully crystalline repeating state. We know it, and again, this goes back to tradition, and this is why you use DSC, for example, as your primary, one of the primary methods, is because you're looking at the heat capacity changes. Because we know amorphous material, when you go through an amorphous change of state, there's a heat capacity change in this glass or amorphous state, much like typical window glass. So that's really where we're focusing, and I highly recommend taking a look at some of the discussions that are currently going on in the literature in order to define exactly why we're getting this boson peak and what is the nature of this random or order disorder in a crystalline or semi-crystalline matrix. In the next slide here, and this is just a nice quick example, and I'll show you, and hopefully it helps clarify. You can see this long amorphous boson peak, fairly undefined, as you're approaching zero, or the laser line. And in this particular case, I cleaned this up because we were following these changes that were occurring with one of our compounds. If you were to monitor, let's say, at 30 reciprocal centimeters, I'm not going to see a difference, and that's what's shown below. But if I focus in at 20 or at 40, I can either monitor the crystalline formation, which means the disappearance of the amorphous, or the disappearance of the amorphous in the process. So it depends which one I want to monitor. And in my particular case, I would monitor both, since we can monitor a number of wavelengths simultaneously, build a model off of it, and you could see over a period of time, we're actually going from amorphous to crystalline material depending on the temperature and the conditions that we have it under in a slurry, for example. So these are some of the stages that can occur, and we're going to monitor them in order to make a nice, desirable crystalline material. This is from an early study where we had a mixture of amorphous material and crystalline material. We did it on a weight basis, so we could actually look at the differences going from 10 to 70% amorphous in crystalline API. So this is taking our primary method results using solid state NMR and DSC and powder X-ray. So we've confirmed what's amorphous and what's solid, and then mix the material as best you can, take multiple spectra and plot that out. And you can actually come up with a PLS model to differentiate and follow a process as it's going from going through a transformation from amorphous to crystalline. Of course, the caveat to that is it has to go to the right crystal form that you're driving for. So I have examples of that. Now, this is from earlier work. This work was done by Michela Raglione under the direction of Peter Larkin a number of years ago. The reference is enclosed there. And I've included the higher frequency ranges along with the low frequency. And if you notice the scale on the left-hand side versus the right-hand side, it's easy to differentiate the amorphous material and the intensity of the crystalline material. So we've got crystalline material, pure amorphous material, and then a 50-50 mixture of the two. So it shows you the enhanced sensitivity. In this particular case, you can see some of a spectral band in the carbonyl region due to the amorphous material. It's much better to look at that low frequency range, and that's where we focused on. And what was nice here is this was a transformation, and this is a known compound, indomethacin. It's easy to make the amorphous material by melting it and then flash freezing it, taking it right down to liquid nitrogen, or in this case dry ice temperatures, and you get a nice amorphous material. And over many, many hours, it converts to a crystalline matrix, and that's what we wanted to follow. And this was a good case study for us to see the versatility of using LFR, low frequency Raman. So from 0 to 60 hours, you could see in the low frequency range that broad boson peak begin to dissipate and the crystals begin to grow out of it, specifically at around around 40 and 60 reciprocal centimeters. So there's really a nice way of tracking this. As a result, we can then do a model and build either taking the crystalline peak at 70 or the amorphous band at 21. And if you remember, that was a nice, fairly clean region where it was just amorphous, and model that and watch that conversion over time. We were very fortunate because it took a long time. So you can actually run this over many days. It occupied instrument time, but it gave us good insight into this process and change and a nice way to build a model. And that helps with our experience as we're going forward and applying this to new compounds. Now, there's also a temperature control studies that we've done converting various forms, in this case from form A to B. And this is what's typical for us. In this study, the crystallization was done at 57 degrees C, so it's actually quite warm. We seeded it with a mixture of unfully characterized crystalline forms of this API. And you can see it forms a slurry. We've got a crystalline matrix. We're monitoring it by low frequency. We're watching it by time. I think this was around 37, I believe, reciprocal centimeters. The point is we're watching this change in the low frequency. You let it go long enough, and there's a secondary transition. So this was what indicated to us, based on indomethacin, and what you see here, is that we have to make sure that we do long enough studies to ensure the crystalline matrix. And I have another example where we saw these changes that were unexpected over time. So it's a nice example for us to monitor for the future. Now, there are a number of changes that we could monitor in this case. There's a number of spectral bands. Because of the concentration here, and the spectrum was fairly noisy, it was a very dilute slurry that we had. So it was a fairly low concentration, so we had to up the spectral collection time. It's usually very fast, it's 3 minutes. We tried to boost the concentration in this case. We were running around 7 minutes, 6 minutes to collect data at a very low concentration. Once we got the concentration up a little bit higher, then we started looking at some of the noise and eliminating that randomness. Since it is a slurry mixing and there's different sized crystals that are formed in it, and obviously it would have to be milled later, but you can see using first derivative, we can easily clarify what peaks we should monitor. So many times we will do a pre-processing of data by taking the first or second derivative to clean the spectra up and narrow down what spectral bands we want to look at. Here's some of the key spectral shifts when we decrease the temperature to 47 degrees C. We dropped it by 10 degrees. We see a really nice shift here from 29, 36 reciprocal centimeters. It gives us a handle on, well, let's monitor this and watch the change in form over time. In addition, at 47 degrees, there are other changes that we could monitor a little bit further downfield, but we have our choice of which one is going to be the best to model. Now, at that 47 degrees, we did some more crystallization studies, and we found an unknown form, something that hadn't been observed based on the temperature that we first ran it at. So we did initial seeding, and for some reason it started to form an unknown form, and this is something that we had to then characterize, and then it went to the desired form after 22 hours to form form B. That was what we wanted, but we saw that, yeah, there's a transition here. This is why the importance of doing these long-term studies to ensure stability, partly because if you're in a manufacturing environment, you may not be able to process that data very quickly and dry it. So it may remain in contact with solution for an extended period of time, where it's then loaded into either a filter dryer, a mixing dryer, or you may need to jet mill it as a slurry if you wish, and all of that is exposure, and that might cause a change in the crystal form. So here's a way, if you're going to do a jet mill, to check ahead of time any possible transformation. Likewise, you can take the samples from the jet mill or put it in line and monitor it during the process to ensure that you haven't made any changes during your reaction. So that's important for us in order to get to good product. We started to drop the temperature even lower. Takes less time to crystallize, so it's much more efficient, and looking at some of the other spectral changes that occur, and this one happens to be at 43 degrees centigrade. And that's where we're able to stabilize it, monitor it, and make sure that we're going to one product over a period of time, shortening the time, and we're ensured of quality. Now, one thing I alluded to is we can do this inline, and that's an advantage that we have with Raman spectroscopy. We have the instruments so that we can do it inline with a probe. We can stick the probe in a small vessel in a laboratory benchtop and do studies. We can take that into a plant through fiber optic cable and monitor what's going on in whether it's a jet mill or your crystallization slurry mix. Likewise, we can pull a sample and shoot right through a vial, a clear glass jar, borosilicate container, and monitor the crystallization that's going on there. So we have very small scale setups in 10, 15 milliliter vials that we can monitor during mixing and monitor the crystallization, or we can take it up to hundreds of liters if you need that size to monitor. This is another study, and this was another very dilute reaction, and it was kind of a lesson learned here for the scientists that were working on, engineers. I put in the times here because the times are actually very important. If many times you want to start a reaction early in the morning, and by the end of the day, before you go home, whether it's 6, 7, 8 hours, 9 hours, 10 hours later, pull a sample, stop it, you're done. Your other option is to start it at the end of the day. You may have constraints, other work going on, and sometimes you'll get to that point, or you'll run a reaction and then you want to do the crystallization. It starts late in the day. The point here is that after 1 hour, you already have a crystal form, but as you let it sit much longer, you get a secondary transition. So if you were to monitor this fairly quickly after, let's say, hour 3, 4, 5, you have one form. If you wait till about 14 hours later, you come in early the next morning after starting it late in the day, you might have a mixture, and that's what the chemist saw. If they come in later in the day, let's say 10, 11 o'clock, they're already 16, 17 hours, 18 hours after they started it, they have a second form. So this is a way we could do this without monitoring constantly for 24 hours or 48 hours, put the probe in, or do non-contact right through a vial, let it stir, watch the crystallization, walk away, and come back with your data, process it later, and note that you have changes. Now you can go back and customize. You can shorten it, you can lengthen it, you can isolate those different crystals, and then characterize the different forms. That helps with the patentability, that helps with your proprietary concerns. You also have to be able to file all the possibilities, and you need to test them for bioavailability, solubility, and so on. So it's a great way, a great tool to get into the intricacies of crystallization. Now, we also look at, obviously, rate studies, as I mentioned. This happens to be a case where we're doing actually very fast cooling at 20 degrees a minute. And we were hoping that we would only have one transition. As it turns out, when we started taking a closer look at the data, there were actually some transitions occurring during the crystallization process, even at fast cooling. So should we go with fast or slow cooling? So this is where the QBD steps in again. Well, let's run it at different temperatures. So you do different temperature studies to look at what form, how long it takes, and what is the quality of the material. The quality is what really impacted here also. So taking an even closer look, you could see this nice shift from 29 to 32. So we could build a model off this and also monitor and know what mixture or what level of different crystal forms we have during the process. If we took this down to 1 minute cooling rate, it ended up being a very long cooling process, long crystallization process, but we got very nice homogeneous crystal forms. That helped us out significantly. As it turns out, if we did a faster cooling rate, anywhere from about 15, 16 degrees and faster, the best we can get was 96% purity. If we took it at a very slow cooling rate, under 4 degrees a minute, we can end up with greater than 99% purity of that crystal form. And obviously, we would pull the samples and submit them for solid state NMR and DSE, and you'll eventually get your results and you'll confirm it. But now we already have the ability to be able to have some control in situ. So that helps us out significantly in getting our work done. Now, I'll finish off here with going with identifications of crystal forms in a slurry. I've cleaned this up so we've got just specific crystals that we were isolated. There was a methanolate form of this API. The desired form is N3. That's the crystallographer. Crystallographers always come up with these names for it. We also have P12 and M2. N3 was the desirable form due to its bioavailability. So that was the primary target in this particular case. So what I'm showing here is a slurry mixture containing N3 and P12, and again, the standards for N3 and isolated P12. So that shows you what our target is. We know what we're focusing in on. We're going to focus in on this region here. We can also use a region up top. So this is very, very important for us. If we do that slurry control, do a good seeding with N3 crystals, we can end up with a nice N3 form. And so here we have a start of crystallization. The crystals grow. We've got N3. You can overlay it. Success. We've gotten what we wanted. We just know that we have to have a certain amount of seeding percentage, 4% of N3 seeds that have already been determined to be pure. Seed that at the right time, the right temperature, the right mixing rate, and sure enough, we can get quality N3 form. So, in summary, low frequency, rapid, very flexible, non-destructive. If you're doing a 4, 5, 6, 10 milliliter crystallization or microcrystallization in a well plate, you can get the spectra with a low frequency Raman. And that's a big bang for the buck because now you can do a lot of screening work, a lot of characterization, a lot of information with just a very small amount of material. You don't have to run a large batch. Very, very powerful tool. In addition to that, we can look and differentiate between the crystalline form and amorphous form. And like I said, that's really important for us to know how much amorphous or if any is present. And we don't want it present. And another big advantage, we can do it inline or offline. I could develop it offline through a non-contact microscope objective, through a probe that doesn't go into the sample, and then take that same method once I build it, and if it's done properly, apply that inline to a probe system in a much larger vessel. That gives us maximum power, maximum control, maximum yield. That concludes this section of the talk, and I'm going to pass this back over to Stephen. Thank you very much for your attention.
Thank you, Dr. Wasylyk. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar. Please feel free to send your questions in at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. James Carriere is currently the product line manager for Coherent's THZ-Raman product line. He has spent the last 8 years as Director of Business Development at Ondax, where he focused on developing the THZ-Raman application space and building the THZ-Raman product platform that has gained wide adoption within the pharmaceutical industry. His responsibilities range from guiding the product design and applications development to sales and marketing, and the establishment and management of global distribution and OEM partnerships. He has a BSc in physics from McGill University and an MS and PhD in optical sciences from the University of Arizona. I will now be handing over to our second presenter. Welcome, Dr. Carriere.
Thank you, Stephen, and thanks to John and all the attendees. I think that was an excellent setup for everything that I'd like to present here. So I'd like to now shift the presentation from some of the applications to how do you actually collect this information and what equipment do you need to access these low-frequency Raman systems, or as we call it, THz-Raman, because the low-frequency portion of the Raman spectrum actually is corollary to or correlates to the terahertz absorption spectroscopy. So the same vibrational energies are achieved from Raman in this low-frequency portion of the spectrum. So just as an overview, I'd like to present our various different terahertz-Raman systems and applications, some of the various sample interface accessories that John was already referring to, show some high-speed mapping capabilities that we've developed, as well as high-throughput screening capability and the data visualization and analysis associated with that. And then we'll finish with the Q&A session. So terahertz-Raman, of course, is useful for the pharmaceutical industry, but it's also very common in many other industries as well, from industrial polymers to the semiconductor materials industry and various others. So the main thing to keep in mind is that terahertz-Raman can be useful for basically any process that's concerned about the structural properties of the materials. So for the examples that John presented, you're looking at crystalline versus amorphous, but anything that where the structural properties are changing, you will see these changes happening in this low-frequency portion of the spectrum. So what are some ways that you're able to add this capability to a Raman system? One is that we have various different modular upgrade systems that we can offer. All of these systems you can think of as basically fiber optic probes that have an optical fiber coming from the modular system that can connect into any existing spectrometer. So if you already have a Raman system, it's easy to upgrade it to having terahertz-Raman or low-frequency Raman capability by purchasing one of these individual modules and connecting it into your existing system. They all contain the same lasers and filters to produce the Raman spectrum, and then just a simple optical fiber output. So we have our benchtop type system, which you can see in the lower left-hand corner, that is roughly about the size of a ream of paper. And again, it has a number of different sample accessories that we can use to interface with the sample. On the right-hand side of the screen, we have our microscope system that allows you to integrate with various different standard conventional microscopes. And it has an optical switch, a selector switch that allows you to bring the unit in and out of the optical beam path so you can convert between the terahertz-Raman mode versus any other configurations that you might have on your microscope. And then on the bottom right-hand corner, you can see our probe version, which basically now separates the filter probe head from the laser source. And so that allows you to put the probe head into a different environment. As John mentioned, you could then be able to run fiber between the laser and the spectrometer to the probe head. And so you can separate the electrical components from the passive probe head that can go into a volatile environment and makes it very much more versatile. And then, of course, it has a number of different sample accessories. We also do offer complete integrated systems that combine the spectrometer and with our terahertz-Raman modules, as well as, for example, mapping capabilities with a microscope, translation stages, integrated software that makes everything easy to use and collect. A good example of this is our WPS system for high-throughput screening with well plates. So as John mentioned, if you wanted to do a microcrystallization experiment, this all-in-one solution could be very, very useful for rapidly detecting and determining a quality process in a high-throughput screening well plate-based environment that can work with basically any standard well plate with a glass bottom up to the 1536 well plate or well configuration. For the modules, we have a variety of different sample accessory options. So depending on what's going to be optimum for your specific interface for your samples. So you can use our vial or tablet holder in the top right-hand corner for use with, say, HPLC or scintillation vials. Below that, we have our non-contact optic. So for, as John was mentioning, looking in through a glass reaction vessel, you could be able to focus with a longer working distance through the glass window. And that can be oriented in various different directions, whether you know, depending on how you want to work with it, and be able to use various different objectives or that can be interchanged quite easily. Below that, we have the contact immersion probe tip to be able to immerse it directly into a reaction vessel or be able to come in direct contact with the system. So the difference between the non-contact versus the probe tip is really whether you're wanting to have a longer working distance or a shorter working distance to interact with the sample. And then on the bottom left side, we have an accessory to mount the probe to a microscope as well, adding even more versatility to the probe system so that you can be able to easily connect all the various different sample interfaces, put it into the reaction environment for processing, and then still be able to connect it to a microscope and be able to do micro-Raman analysis. And beside that, we have our transmission Raman adapter as well. So for the same probe unit, it can be a backscatter Raman instrument or a transmission instrument. So just a number of examples of various different sample accessory options that we have show the versatility of this technology and how easy it is to incorporate it into your solutions. So how do we actually do this? The simplest thing is it's as simple as disconnect the existing fiber from your spectrometer if you already have one, then connect the collection fiber from the THz-Raman system to your spectrometer, and collect data using the spectrometer as you normally would. So you don't need to have special software to gain access to this. You don't need to change any of your standard processes. You'll still be able to collect everything. It's just simply adding a new probe capability effectively to your system that allows you to gain access to all this special information that John mentioned. Now, in some instances, we need to shift the spectral, the wavelength of the excitation laser to access this portion of the spectrum for some spectrometers. In that case, depending on the software that's used, you may need to use a correction algorithm to shift everything back to matching what you're used to, but we can provide this using our SpectraShift software program. So just an example of how we can connect that. Here shows one of our probe units connected to a Kaiser RXN system, even with another adapter that allows you to use the Kaiser non-contact optic. In this case, it's focusing onto a reaction vessel, looking at a reaction of anhydrous theophylline going through a hydration experiment. And so with the data on the right, you can see how over a period of just 100 seconds, you can see the sample go from anhydrous to monohydrate forms. So, and all of this is clearly evident in the low frequency portion of the spectrum. Of course, with all these different pharmaceutical APIs, most every one of them is polymorphic in nature. So here's 3 different examples showing the various different polymorphic forms. You can see with the system, we're able to collect the entire chemical fingerprint region plus the low frequency portion of the spectrum. It's really just limited by the spectrometer that you connect it to. But you can clearly see how strong the signals are in this low frequency region. As John mentioned, you can get an order of magnitude boost by looking at these low frequency or THz-Raman modes. And if we zoom in closer into the THz-Raman low frequency spectrum, this is where you can clearly see the differences between the different polymorphic forms. As you change the structural form, you see these corresponding changes happen. And for example, with the indomethacin, you have the highly crystalline gamma form versus the amorphous alpha form. And the presence of those strong, sharp bands clearly show you the crystallinity of the material. So this can be a good crystallinity meter effectively by taking advantage of the low frequency Raman spectrum. Similarly, if we were to look at a microscope configuration attached to a THZ-Raman workstation, we're able to use Micro-Raman to look at the high spatial resolution imaging and mapping for a co-crystalline application and be able to see the formation of co-crystals. In this case, we're seeing in red a physical mixture of caffeine and 2-benzoic acid, but when you form that co-crystalline bond, you can then shift to the blue curve, which gives you this co-crystalline complex. So as you form that crystalline bond, it changes the vibrational degrees of freedom in this low frequency portion of the spectrum, and that's what produces these corresponding shifts in the spectrum that are easy to identify. Another accessory we have is our floodlight. So for instances where you may have some spatial distribution of your sample and want to get an idea of exactly what sort of mixture you have or do some quantification analysis, you may want to to get an idea over a larger surface area. So in this case, when you might have with a single focused spot size of about 20 microns, it could be very, very sensitive to the spatial location. But by adding this floodlight accessory, we're able to increase the effective diameter of the spot to about 2 millimeters. And so while you can see spatial variations at individual locations, 1, 2, and 3 in the spectra on the bottom, when you turn on the floodlight mode, you see just the average over the area that is very, very repeatable, and that allows you to do this quantification much more effectively. Beyond the pharmaceutical space, polymers are a very hot area as well for looking at these structural changes. In this case, high-density polyethylene, which is used for various things from plastic bags and bottles to pipes and housing wrap. If you change the lamellar thickness of the polymer layers, you will change the corresponding structural properties of the polymer. When looking at the chemical fingerprint region, they all look basically identical, but in the low-frequency portion of the spectrum, they can be very, very different. So it's not just a process tool for pharmaceuticals; it can be processed for many, many different types of applications as well. The transmission Raman accessory is a little bit different now, where normally we would connect the source fiber to the back of our probe head, and on the same side as the collection fiber for backscattered Raman capability. With the transmission accessory attached, we now connect the fiber to the other side, to the right-hand side of the sample. And so effectively you have the source and collection fibers staring at each other with the sample in between. And this allows us to get a volumetric average of the entire material that's present between the two fibers. So in the spectra that's embedded in the slide here, you can see the blue spectrum is the backscatter case for looking at a standard coded Advil tablet, and you'd be able to see the backscatter is coming mainly from the tablet coating itself. But when you move to transmission mode, you're able to see now the entire volumetric content of the material. So you're able to see the underlying API with a much greatly reduced effect from the tablet coating based on the volume contribution to the tablet. In this case now, you can see the terahertz or the TR probe system attached to a microscope, one of the thermo DXR Raman microscopes, using our microscope adapter. So this allows you to, again, be able to use the same probe device and be able to do micro-Raman mapping with it as well. So it makes it very easy to integrate. Again, you still have just the fiber connection to the rest of the spectrometer. You can select when you want to have the instrument put into or out of the beam path, and it's compatible with all sorts of different applications. So one of those, if we wanted to say map a typical tablet, we can look at various different APIs, amorphous versus crystalline components, plus the excipients that might be present as well. And you can see that one of the advantages of the low frequency region is that the excipients tend to not have very much information present, so they do not act as a confusing factor for the various different APIs. So it allows us to focus really in on just the amorphous and crystalline content themselves. And here we can see two different compositional tablets that were formed. On the left-hand side, we had a 50-50 mixture of crystalline versus amorphous API. On the right-hand side, we have 90% crystalline, 10% amorphous. But in the mapping mode, we're able to clearly be able to identify all the different regions where you have the crystalline versus amorphous forms, and be able to get a good validation of what the content is for each in the tablet, to determine the best possible process that you're wanting to produce. So now I'd like to move on a little bit to our high throughput screening tools. So again, it's compatible with any standard well plate that has a glass bottom. We do a bottom illumination to be able to easily determine the exact plane where the sample is located. But otherwise, it's a completely integrated system combining all of the aspects of the microscope, the translation stages, the spectrometer, and the THz-Raman module to collect all this data. So we have an intuitive graphical interface for selecting which wells you'd want to measure, and we have various different scanning patterns that you can use to collect data from within the wells. It incorporates polarized light microscopy imaging, so you can be able to visualize the specific portion of the well that you're looking at, with various different objective lens options, so that you can choose what magnification you'd want to use to measure everything. Plus a live Raman imaging mode as well, so that you can be able to click on a specific portion of the visual image and immediately see the corresponding Raman spectrum. So if you want to then collect all that data, you can easily set up all the wells that you want to measure, the corresponding measurement patterns, collect your entire data set, and then with our analysis software be able to identify, even in a sparsely populated well, where the specific crystalline or material components are located within each well for the entire well plate, and be able to then do a principal component analysis and clustering to determine what specific species are located in each specific portion within the entire well plate. And then you can even zoom into a specific portion of the well and see what sort of distribution might be present within that well, and be able to get all the data from that at one time. Similarly, it's not limited to just well plates. For the high throughput screening, you can actually do mapping with the well plate system as well. In this case, with an Eccentric Tablet, you can load this in with a collection plate, do your standard data collection, chemometric analysis, and then visualize the output. So, and then be able to determine where you might have the acetaminophen, caffeine, and aspirin components within the Eccentric Tablet. And if we look at the individual spectra that are present, you can pull up each individual data point and see the corresponding spectrum that comes along with this. So it makes it very easy to collect all of the data, do the analysis, and visualize both the spectra and how it relates to everything else within the data set, all within one system. Extending that to a fast mapping capability here. So again, with the same tool, we can be able to, within about 35 minutes, be able to capture over 200,000 spectra for this Eccentric Tablet, and be able to identify all of the different spectral locations for the various different species within the tablet. Now, part of the reason why we can do this is because we have such strong signals coming from the low frequency portion of the spectrum. So when you do the principal component analysis and clustering with taking advantage of the low frequency portion and anti-stokes from -200 up to about 2,000 wave numbers to include the entire chemical fingerprint region, it makes it very easy to identify all the different components that are found within the tablet. So you can clearly see the regions which would correspond to acetaminophen in red, aspirin in blue, and caffeine in yellow. But if we were to limit the data to to only the fingerprint region greater than 200 wave numbers, scanning so quickly, we then lose the weaker information coming from the caffeine portion of it, because the signal-to-noise ratio has now dropped significantly. So you wouldn't be able to scan as quickly using a conventional Raman instrument that does not have access to the low-frequency Raman portion of the spectrum. So this is a big advantage for allowing you to mapping and screening much faster with having the low-frequency capability. So I guess the big question is, what do we need to add THz-Raman capability to your system? Just contact us. Let us know what you currently have, and in terms of equipment, and what you're trying to do. We have solutions that can meet basically every need. If you already have a spectrometer, let us know the make and model, what wavelength you want to work with. If you want to work in a microscope configuration, what microscope you'd have, or any other special requirements you might need. We're here to help. And then, of course, let us know what type of samples you need to measure. As you've seen, we've got a number of different sample interface options, from HPLC vials to bulk interfacing. So really, anything that you need to do, we can come up with. And if there's something that we haven't created yet, that's how we come up with the new sample interface capabilities. We want to hear from you. So just to take away from all of this, I'd like everybody to understand that the structural information that you are able to collect with the THz-Raman, the full capability accessing the low frequency portion of the spectrum, is really critical for a variety of applications. Anything where you're wanting to access the structural information about the materials, whether it's polymorphic form, crystallinity, changes in phase, or hydration, any of these things will affect the low frequency portion of the spectrum first and foremost. And so that allows you, having this capability can really expand your understanding of the system. So I would encourage you to come visit our website, learn more about the tools that we have available, contact your local Coherent sales representative or myself, and we'll be happy to work with you to help you gain access to this information. Thank you very much.
Thank you, Dr. Carriere. Now we will begin the questions and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the question tab located directly below your webinar screen. So now our first question. In the case of non-contact Raman spectroscopy, what are the limitations and focal distance? For you, Dr. Wasylyk?
Limitations on the focal distance? It depends on, and as James had mentioned, you can actually do essentially standoff Raman. In other words, you can have your probe or objective tuned towards just the surface, or any distance off of it, several millimeters, several centimeters if you want. And obviously in national defense, they do it in terms of meters for standoff Raman. Obviously, there's going to be some dispersion there of signal, but typically we will have it so that we could go through fairly thick glass vials, so we can collect the spectra without having to insert a probe and for a quick analysis. It's especially important if it's something that's a toxic material. You're worried about exposure to the solvent, the reagent, the chemical. You can avoid all that.
Thank you. Our next question is, when analyzing a crystallization process in situ with a probe, how do you avoid probe fouling? Again, would you like to answer this, Dr. Wasylyk?
Yeah, a lot of that has to do with the placement of the probe in your vessel. If you're going to do small scale, there's usually not an issue because you can seat your probe right near the agitator, as close as possible, obviously without contacting it. So you have a lot of shear, and you're going to rely on that shear to help keep it clean, so to speak. If you were in a manufacturing environment, you have to make sure it's near the impeller but not behind any baffles you have where you can have small eddies or microcurrents that'll just circulate. The exchange is a lot slower and there's a greater chance for fouling of the probe. So that's really the only way, is you're going to rely on the nature of the mixing to help clean the probe or keep the probe in a good state.
Thank you. And our next question. Are you able to transfer small-scale studies, say non-contact in vial, to large-scale production? Again, Dr. Wasylyk?
Yes, we are. It depends on the models that you're able to build, and we've fortunately, the team here, Ming Wang, Bob Wethman, have done a stellar job at creating methods that are robust. We do a lot of testing of the methods, and since the instruments are very, very well matched, and what we're able to do is build simple chemometric models that incorporate, for example, ratios. You can build a PLS model, you can go to town and make it very complex. We find that simple ratios and a simple PLS model works very well because you're normalizing it so you can address those concentration differences, because each batch, even though you're following a recipe, are going to have some slight variations in it. That's an entirely another discussion or talk, but there are ways to make really robust methods that enable us to be able to transfer from small vessels to large, and we've been doing this actually for a number of years.
Thank you. Our next question. How can we separate specific protein in media among different protein that also interfere with purification? Dr. Carriere, would you like to answer this first?
Sure. So this is an interesting problem. So trying to look at proteins is a bit more of a challenge than some of the small molecule applications. Effectively, as you increase the molecular size, you gain more and more modes in the low frequency portion of the spectrum. So you really have to do much more of an analysis of the corresponding changes, because all these modes will start to overlap with each other. It's something where if you're looking at specific changes in the proteins, you can do the similar analysis to what John showed looking at the crystalline versus amorphous, where you could find this corresponding region that would have a specific band or bump that would be an indication of the protein that you're looking for, as opposed to the background media that can be present in the mixture. But it's certainly a complicated analysis that you'd have to do. It's not necessarily something that's trivial in nature. John, do you have any other things to add to that?
No, I think you covered it.
Okay.
Yeah, that's a challenge. We've only made minimal attempts to look at proteins in low frequency, and that's actually on our docket to do. It's a matter of finding the time, but that is something I really want to spend more time on. It's trying to fit it into the daily schedule. Yeah, I think it's going to mean nighttime and weekend work.
Thank you. Our next question. Can you quantify residual crystallinity in amorphous solid dispersions, or ASD? And if so, what LOQs can you obtain? Again, Dr. Carriere, would you like to answer this first?
Well, I mean, again, this is kind of sample specific. Effectively, the limits are really going to depend on how much difference you see in the spectra. Certainly, as John showed, you can see basically all amorphous material will have kind of these broad spectral features, and highly crystalline materials tend to have these strong, sharp bands in the low frequency portion of the spectrum. So it's generally when you have a combination of crystalline and amorphous materials, you can usually find a region where you'll have some sort of a quiet range that's present in the crystalline portion, and you'd still have a signal of the amorphous region, as John had identified. So in terms of the limits for what you'd be able to detect, it really kind of depends on the separation between these various different modes. Certainly, we've done some testing and been able to indicate that you can get down to around the 1% range or below, but that is highly sample specific, so it really depends.
Again, John, do you have anything to add?
No, that was actually the perfect answer because it depends on how specific and the separation you have.
Thank you then. Our next question. How does spectra look like if protein is in the correct form, or dentured form, or even in misfolded form? Who'd like to answer this one? John, would you like to answer this?
I can't because I haven't done enough work with proteins yet. As I said, that's on our docket to do. Matter of fact, I'm replying to somebody that has already brought this up to my attention, and yeah, I think maybe we'll be collaborating on this soon.
James, do you have anything to add?
Yeah, this is a little bit difficult as well. I think the best thing to do would be to contact us. Another thing I didn't mention is that we do have a capability to measure customer samples in our facilities. So we're happy to work with you and be able to measure customer samples and prove out things that we don't necessarily have experience directly with, to be able to answer some of these questions in greater detail.
There is actually some literature out there on using terahertz and protein changes and conformational changes, but hands-on experience, that's hopefully going to come very soon.
Yeah, I think the problem that comes back with what John mentioned earlier, that access to this low-frequency portion of the spectrum was so difficult for so long that people didn't really work with it. So the understanding of exactly how all the differences are changed, this is really kind of like the new frontier of Raman spectroscopy that we're developing here. So it's just an exciting field of research that for everyone involved.
Well, thank you both for your answers, and thank you, audience members, for your questions. I think that's all the questions we have time for. But before I finish the webinar, I'd like to ask our presenters, John and James, if they have any closing remarks. John, do you have anything you'd like to finish with?
The only thing I will mention is that there are a number of conferences around the United States and international. One that comes to mind quickly, it's also near and dear to my heart, is the SCIEX conference, sponsored by the FACS organization, Federation of Analog Chemistry and Spectroscopy Societies. That's being held in Palm Springs in October 13th through 18th this year. You can still submit papers, you can attend, and there will be sessions on terahertz, low-frequency Raman, and general vibrational spectroscopy. And I know James is going to be there.
And James, would you like to add anything?
Yes, that's a great conference. We'd love to have you show up. Please don't even wait for the conference that takes place in the fall. Reach out and contact me at any time. I'd love to hear your ideas for how you'd like to implement the low-frequency Raman capability. So please, you take advantage of contacting us through the website or emailing me directly. I'm all ears and ready to help.
Well, thank you both. I would like to take this opportunity to thank our presenters once more. That was John Wasylyk, Senior Principal Scientist at Bristol-Myers Squibb Company, and James Carriere, Product Line Manager for Coherent's THz-Raman product line, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.